Hello, everyone. Welcome to Examining Evolution. I'm so excited to have SFT, Standing for Truth, here with me today. And we're going to talk about endogenous retroviruses. And he's recently come out with a book called the Endogenous Retrovirus Handbook. And his book is showing why, although endogenous retroviruses are often promoted as evidence for evolution, Donny, your book is showing why that's not the case. Is that right? Donny, your book is showing why
Yes.
that's not the case.
Just one second. I've got your...
Yeah.
We're good. Yes, exactly. It shows why endogenous retroviruses are not what the evolutionary community makes them out to be, like, the single best evidence for common ancestry. And it not only refutes ERVs as evidence for common descent, it also provides an alternative explanation, a superior model, essentially.
Great. Well, can you start by telling us why are endogenous retroviruses said to be good evidence for evolution?
Absolutely. So, firstly, I appreciate you promoting the book. So I've got 2 copies here. So this is, the copy that most people have, but currently there is like a limited edition version. So the Endogenous Retrovirus Handbook, limited edition, that would be like your most up-to-date version. And, praise the Lord. It was pretty cool. The first week of release for the Endogenous Retrovirus Handbook, we were the #1 release. So, it's a blessing to see that a lot of people are benefiting and profiting from it. As you know, we have an evolution debate challenge series going on for 2022. I think we've done about close to 30 debates now. And I would say at least 75% of them, the top 3 lines of evidence that the evolutionist debaters bring forth always consist of endogenous retroviruses. So that was one reason especially that I thought, okay, the creationist world, the young earth creationist camp, we need, a single comprehensive response to endogenous retroviruses as evidence for evolution, where everything's kind of compiled into one. And so, before I released it, I did have some feedback from some professional creationists as well, who were quite happy actually with the arguments and responses in it. But basically, endogenous retroviruses, Rebekah, are these small pieces of DNA that are found within the genomes of many kinds of creatures, especially in invertebrates. And they resemble viral genetic material, right? And they are found, let's say, in primates, or our so-called closest cousin, the chimpanzee. So they're found, these viral genetic sequences are found in corresponding locations in the genomes of humans and chimpanzees. And when they say these sequences fall out into nested hierarchical patterns in terms of their distribution, what that means is we would share more of these sequences with the chimpanzee than we do with, a more, what the evolutionists would say, distant common ancestor, like an old world monkey or a lemur. So there's this hierarchical distribution where we share more with the chimpanzee than we do with the gorilla, than we do with the orangutan. And the further we go down the line, we're going to share less with, a dog or another form of life. So the evolutionists believe that these shared endogenous retroviral-like sequences that form nested hierarchical patterns are good evidence for common descent, because they'll say the reason why we share these viral genetic sequences is because we inherited them from a common ancestor. And of course, it goes a lot deeper than that as well. But for sake of time, I'll call it quits there on that question.
Okay. So, are endogenous retroviruses, are they viruses?
That's a good question. So the question I like to ask, Rebekah, is, are these really the ancient remnants of past viral infections that have been passed down vertically over millions of years, right? Or are they created units of DNA function? So anybody familiar with me or my debates, discussions, they'll know that's a question I like to ask. So the question you're asking, are they viruses? That is the key question. Because, and what I'll do is, if it's okay with you, I know I'm a visual learner, so, and I understand a lot of other people are too. I can show a diagram of herbs. Just let me know when my screen is shared and I can go over some of these.
Yep.
Some of these slides.
Your slides are there.
And I see in the chat we've got some of my arch nemesis. One of them, Andrew Cummings. So I just had a 2-hour, informal debate discussion with him last night. And we touched on a lot of the details, actually. For the first hour, we touched on all the details pertaining to what are endogenous retroviruses, right? So right here, endogenous retroviruses, what are they? Once retroviral DNA becomes incorporated into an organism's genome, it is called an endogenous retrovirus. Rebekah, this is because the ERVs originate from within, right? Where your exogenous retroviruses, typically when somebody hears about exogenous retroviruses, they think of the HIV virus, which if somebody's infected with an HIV virus, that virus can be passed on horizontally, but not vertically, as in they're not passed down to, subsequent generations. So again, here, after inserting into the host genome, the endogenous retrovirus can still produce retroviral particles if its DNA is transcribed by the host cell's biochemical machinery, if the ERV infects a germ cell line. So this is where it's key, is if you have one of these exogenous retroviruses that insert itself in the right way in the right spot, okay, let's say a sperm or egg cell, and then fertilization occurs, then it can be inherited from generation to generation. So to the evolutionists, when we look to these sequences, they'll look to it as a historical account or a historical record in terms of evolutionary history, common descent. So that's a little background, that's a little context. So going to your question, are they viruses? Well, evolutionists believe they're viruses because, as you can see here in this specific picture, the way that the ERV sequences, Rebekah, are structured is very similar, if not nearly identical, to these exogenous retroviruses that can be passed on horizontally, like HIV. Okay, so if you look on the slide here, you have what's called the LTRs. So these stand for long terminal repeats, right? So you have host DNA on either side. Then you have the viral genetic material. So long terminal repeats on either end. And in the middle of the ERV sequence, in the middle of these LTR sequences, you have what's called the GAG, POL, and ENV properties. The GAG, POL, and ENV genes. Now, it just so turns out that this is the structure of the ERV, but it's also the structure of retroviruses that you can, be hit with. Obviously, can make you sick, lead to disease, and then therefore you can pass it on to other people. So, that would be one of the few reasons why evolutionists would believe that these ERV sequences then must be the ancient relics, I guess you could say, genomic fossils of past retroviral infections, right? So
now, is this
something
that we've observed? I'm sorry, I just wanted to ask this before we move on. Is this just something that's assumed? Is it assumed or is it observed?
So that's a good question. So what's called pre-existing or fixed ERV sequences. For example, fixed just means to be stuck in place. So that means, for the most part, every single human being has these retroviral sequences, okay? So they would be fixed and they would be pre-existing, as in they were never observed, right? So that's a good question that you asked. They were never observed to be inserted. And throughout my book, I back up everything I'm saying with the technical literature, and they would admit that, yeah, for the most part, the majority, that's usually what they say, Rebekah, the majority. You can see this in chapter 1 of my book called The Details. The majority of ERV sequences happened millions of years ago, the insertions. And so we're just looking at the viral genetic material as they are, right, in their positions within the genome today. So it is an assumption that these are the ancient remnants of past viral infections. That being said, we do have what's called unfixed ERV sequences. And so, those look like they could certainly be the remnants of exogenous retroviral infections. But why this is important is because of one of their functions, the pre-existing ERVs. So if you look at, and we'll get into this because this is one of the main objections from the evolutionary camp, Rebekah. So important questions to answer. Why are there ERV sequences shared between the genomes of organisms? 2, if viral-like sequences in the genomes of organisms are functional, so that's my position in the book, is that the reason why we share these in corresponding locations, let's say between humans and chimps, is due to common design. They're created units of DNA function. Therefore, we would predict function, but critical function, essential function. And that's what I want to go over soon. But the second question is, if they are functional, and we'll get into that, Rebekah, I think that's important. The second question is, if viral-like sequences in the genomes of organisms are functional, why do they bear similarity to viral genetic material, right? The first question you asked, why do they look identical to exogenous retroviruses if they aren't really the ancient remnants of exogenous retroviral infection? Actually, before I move on, did you have anything to clarify there? Any kind of follow-up questions?
No, we can continue.
Okay. Especially because the questions you're asking right now require a lot of context because the evolutionists, they're going to say, Standing for Truth just admitted that, there are true endogenization events, right? Well, this is why I should start with this paper. Okay, we'll start with this function. This is one of the more fascinating functions, Rebekah, and I'd wonder if you're familiar with this one. I think you are from watching my videos and reading some of the book. So here's a paper from 2021, but in the book I have dozens of papers that touch on this exact same function. So notice this: switching sides, how endogenous retroviruses protect us from viral infections. One thing I want to note is that when you read a lot of these technical papers, they almost sound like an intelligent design article, right? Notice this: long disregarded as junk DNA or genomic dark matter. That's what they've always assumed, that these, this viral genetic material found all throughout the genomes of living organisms, they've always assumed they were junk, non-functional evolutionary leftovers, right? And they admit this here. Endogenous retroviruses, ERVs, have turned out to represent what? Important components of the antiviral immune system. Now, they function in embryological development. They function in determining cell types, gene expression, gene regulation, tumor suppression. Okay, just a few at the top of my head. But this is one of the more fascinating ones, Rebekah. So they not only regulate cellular immune activation, but may even directly target invading viral pathogens. One focus will be on recent advances in the role of ERVs as regulators of antiviral gene expression. So in other words, they act as antiviral protectors. You know how your computer, you have it built with what? Antiviral programs, maybe Norton antivirus, something like that, that would detect viruses and get rid of them before they can be damaging, right? Well, that's what these ERV sequences, that's one of their functions, is that if we're invaded with exogenous retroviruses that are looking to infect us, right, infect the host, these ERV sequences act as a protective barrier. Like, you got the president of the United States and then you got his bodyguards, right? His group of bodyguards. Well, our bodyguards in the genome are these ERV sequences, and they can fight off these viral infections And we're constantly bombarded every single day. You wake up, you go outside, you drink water, you eat food. You're always being attacked by viruses, bacteria, pathogens, things like this. So this is good design. Anybody who first assumes intelligent design understands that, yes, a good designer is going to front load its creation with many functional DNA elements, with some of them being protectors from invaders from the outside, right? But here's the thing that's key, is the fact that their function, Rebekah, is predicated on their similarities to exogenous retroviruses, because they carry out this job in many different ways. But one of them is through what's called viral mimicry, right? And through this mechanism, they can actually fight off the viruses that are invading the host and prevent the possible endogenization of these exogenous retroviruses. It's kind of like this, because I know a lot of this is technical. If you were like an FBI agent, okay, and then you were hired to, you see a lot of these cool movies that people might know of, like Goodfellas, The Departed, Godfather, things like that. You'll have an FBI agent infiltrate the mob. Well, they can't infiltrate the mob with their FBI suit on and their badge. Hey, I'm Donnie, I work for the FBI. No, they got to play the part, right? Does that make sense? They got to look the part in order to infiltrate, gather information, do their job, or else it's going to be useless. They'll be detected and killed right away. These ERV sequences, it's the same thing. They need to look like that which they're fighting in order to do their job. So the evolutionists say, okay, why do these ERV sequences, why are they built and structured with the LTRs, the long terminal repeats, the GAG, the POL, the ENV? Why do they have the enzymes, reverse transcriptase, integrase, right? Why do they bear so much similarity to exogenous retroviruses? And the question is because they have to. One of their many functions requires their similarities to exogenous retroviruses. Last thing I want to say, and then I'd love to get your comments, Rebekah. This is why this context is important in your question about unfixed ERVs. Here's the thing. Think about this. We have many amazingly intricate, what's called DNA repair enzymes. Okay, we're constantly experiencing, we don't even realize it, but through cell replication cycles, we're constantly experiencing DNA breakages. But we have amazingly designed DNA repair enzymes that are detecting those and fixing them. Okay, we have these DNA elements working overtime. We're sitting here just having a discussion, and there's so much regulation going on in our body. But, and I'll just ask you, Rebekah, do these DNA repair enzymes, although they work beautifully, do they do their job perfectly, would you think?
No, because eventually we're going to, our bodies are going to decay and die. So no.
Right. So we accumulate many thousands of mutations during our lifetime. It's primarily why we die, plus the shortening of the telomeres, of course. It's all mutation related. We have our somatic cells. Think of your skin cells. Those mutations are not passed on. But if we have mutations in the reproductive cell lines, well, those could be potentially passed on. So if we didn't have these DNA repair enzymes, Rebekah, we would go extinct in 2 or 3 generations. That's too many mutations. We'd die off. So this is evidence for forward thinking that points us back to the forward thinker, right? Who front loaded the genome with design diversity, functional DNA elements, amazingly designed DNA repair enzymes. But again, that is their job, but we know mutations still happen, right? Mutations happen, they get passed on, they're not all fixed. Well, one of the functions of these ERV sequences are to prevent exogenous retroviral infection and prevent these exogenous retroviruses from infecting the germ cell lines and being passed on vertically to become an endogenous retrovirus. But the question is, just like the DNA repair enzymes in their job, do you think that they do this role? Do you think they do it perfectly?
No.
I can answer it if you don't. I'm not trying to put you on the spot, but the answer is no.
Oh,
They don't.
yeah, I said no. I'm sorry. Maybe my mic is low or something.
No worries, no worries.
Okay.
So
Yeah.
that's the correct answer. Is my screen shared, so I can't see you. But
Okay.
yeah, the answer is no. So occasionally we would expect an exogenous retrovirus to integrate, to infect in the right place, in the right spot, and it may actually bypass our pre-existing, our built-in protectors, the ERV sequences. Okay, so then they're passed on vertically. But here's the thing, and these critics, they dodge my arguments, they dodge my challenges. I have an entire like section in my book here going over my predictions. My predictions would be that the pre-existing fixed ERVs, and then let me know if this makes sense. And of course, I know this is technical, so if you need me to clarify anything, let me know. The fixed ones, the ones that are stuck in place that we all have, that apparently we share with the chimpanzee, those are fixed. They're not unfixed, right? They're pre-existing. They've never been observed to be infected, but they are also functional, essential, critical. What I want to do is take just a really quick detour, and I think you're going to love this. Rebekah, okay, notice this paper. Retroviruses of the human, the recycling of viral genes and the resulting advantages of human hosts during evolution. So if people want to check it out, they can check it out. So HERVs, if you see an H before ERVs, that just means human specific. So it's an endogenous retrovirus, but the H signifies HERV. That's taxonomically restricted. So what that means is humans have this, but chimpanzees don't, orangutans don't, so on and so forth. So they play important roles in physiology, fetal development, and human evolution. That's just lingo for, well, we can't really explain the functions, but everything's evidence for evolution. If the accidental infection, notice this, of a mammalian ancestor by an exogenous retrovirus, again, that's different than the endogenous retrovirus because it's occurring from the outside. Think of HIV. Had it never occurred, right here, this is the important part, the placenta and the mammals that produce it, including humans, we're human, last time I checked, Rebekah, would have never existed. I don't know about you, but I like to exist. I like existing. I enjoy life. If we didn't have these specific classes of retrotransposons, we wouldn't exist. What that means is they're essential. Okay, there's a specific class of retrotransposon in the mouse genome that, see, here's the thing. Knockout tests in humans are unethical for obvious reasons. Okay, I think we can agree on that. Knocking out our genes, that's unethical. So we have mice for that, thank God. So what they can do is they can either turn off or snip out this certain class of retrotransposon in the mouse genome. And you know what happens? The mouse is developing. You snip it out or turn it off, it dies. It stops developing, it dies. All forms of life, in order to even develop in the first place, embryological development, require these retrotransposons. Well, what that means is if we share, yeah, go ahead. Go ahead.
No, I need to interrupt here because I'm getting too lost. I need to get some clarification here. So are you agreeing with this? First of all, are you saying you agree that some exogenous viruses became endogenous retroviruses?
Right.
Are you agreeing with that?
Absolutely.
Okay.
Absolutely.
But you're saying the ones that humans and chimps share, they are ones that according to your proposal, your hypothesis is they were not the type that were exo and became endo, but it was just something that was always present in the genome.
Exactly. So you said it perfectly.
You're saying that's because, okay. And then, so you're saying the reason we know that is because those are the ones that are fixed. And then I need to ask a clarifying question. How do we know which ones are fixed? And how do we know which ones are do you call it floating? What did you call it?
Unfixed.
Oh, okay. Unfixed.
Yeah. So that's a very good question. And there are what's called consensus sequences that we can look at and compare. And we can see if a certain mutation, for example, is only in a certain part of the population. But like, for example, you can have a subgroup. Let's say somewhere in Africa, you have a subpopulation where they've, reproduced within their tribe and not really much from outside. Well, they will accumulate mutations, okay, that can reach fixation, which again just means to be stuck in place. Okay, so within that group, you could have fixed mutational differences where everybody in that group.
Okay, wait, hold on, pause. Sorry to interrupt, but pause.
Yeah, no,
when you say mutation, is that the same thing as an endogenous retrovirus? Like,
good
or
question.
is that a totally separate thing?
Well, I wouldn't say totally separate, but I mean, the evolutionists would look at it as like a genomic fossil. So an endogenous retroviral sequence, they think, is a genomic fossil in the sense that it's a remnant of a past viral infection. So a mutation, which is just an alteration in the nucleotide sequence, right? That is different, but the evolutionists would still say, this is a mistake, although they'll say that sometimes there's beneficial mistakes, right? But the retrovirus is, according to them, a relic of a past insertion. But either way, you can still have fixed versus unfixed. You can have fixed ERVs, you can have unfixed. You can have fixed mutations, unfixed mutations. This is where I want to give a little bit of context, especially for anybody that's new to this.
Okay, so you're saying some humans have all the same stuff, and you're calling that the fixed ones. And then some humans have acquired these ERVs and only in a small population. Is that right?
Right. So for example, every single human being is 99.999% different. We're pretty much all exactly the same. But there are some differences due to mutations that have accumulated due to independent events. Let's say the endogenization event in separate lineages. One example would be, let's think of like a single base substitution or a point mutation, okay? Which is just, changing one nucleotide, one DNA letter to another, substituting it for another. Well, there's a lot of diseases, Rebekah, that are due to a single point mutation, okay? A single point mutation can actually kill an organism, but a single point mutation can actually lead to a disease that's harmful, but is not harmful enough to kill the organism, and so that organism can reproduce and pass on their genes. One example of this is sickle cell anemia. I'm almost 100% certain that the sickle cell anemia allele and disease is due to a single point mutation. Well, here's the thing. We don't all have that mutation. That's not fixed. That's only in like a subpopulation, right, of people. It's actually more prevalent in Africa. So that's an example of a mutation that's not fixed. And the same thing would go for endogenous retroviruses. So anytime you hear, here's an easy way to understand it. Anytime you hear the evolutionists say, we share X number of ERV sequences with chimpanzees, then those are fixed. Because even the evolutionist believes the reason why we share those, think about it. We have a common ancestor 6 to 10 million years ago up here, right?
Okay.
Our common ancestor hypothetically gets hit with retroviral infections. They integrate, they get passed on. Then we have the split. Humans go one way, chimpanzees go one way. Well, what are they taking with them in their separate lines? They're taking the fossil evidence, I guess you could say, for those retroviral sequences. So the evolutionist says, see, we share this one with the chimpanzee. We have this viral DNA in the same locations because we inherited it from a common ancestor, right? Does that make sense? That's why the evolutionists believe.
Yeah.
But those would be fixed because the reason we even share them, we'd have to all have them for the most part. Go ahead, Rebekah.
No, I'm sorry. I'm just trying to interrupt before you go on to another thing so I can make sure I understand. Can you go back to that last slide that you because the one about the quote that says if like if these ancestors hadn't like taken on this virus, then like none of us would have ever existed.
Right here.
That
right
one.
here.
So they're saying, this is from an evolutionary perspective. I'm just trying to get clarification here.
Yeah, no,
This,
take your
from
time.
an evolutionary perspective, it was an accidental infection
Right.
that that caused us to be like, that is what led to us being able to, develop in certain ways physiologically. But are you agreeing with this quote, or are you saying, well, actually it's because God designed us that way and he made the things, and like that's how we got development? Or are you agreeing that there was some kind of accidental infection that led to important things?
That's a good question. I'm glad that you're asking these questions. I am saying that the answer to the question, are these really the ancient remnants of past viral infections, or are they created units of DNA function? The answer is they are created units of DNA function. So God would have front loaded all the original created kinds with various classes of functional DNA units that function in the embryo, function in gene expression, gene regulation, act as antiviral protectors. And so the reason why we would then share, right, at its most basic level, the most basic answer, right, that even a layperson can use against the evolutionists, is the reason why we would then share. Let's say we shared an ERV sequence that functions in the embryo with the chimpanzee. Okay, well, the chimpanzees require embryological development just like we do, and we would then therefore require DNA elements for that. So why do we share those with them? Well, because of common design. I mean, why do all modes of transportation, why do many modes of transportation have 4 tires, windshields, certain height from the ground, an engine, steering wheels? Common design, common purpose, common function. There for a reason. And their answer, Rebekah, to how these ERV sequences became functional, they will either say that the exogenous retrovirus already had the capability of embryological development or determining cell types, which is pure imagination. It's a lie. No, they've never demonstrated this. It's not observed. They can believe it. They can believe that these viruses. Okay, think about this. I find this kind of humorous. So we get invaded with a multitude of exogenous retroviruses, right, that are seeking to harm us essentially and take over. But it turns out that one infects the germ cell line, gets passed on vertically, and then ends up taking on a job that what? Fights off that which it used to be, right? Protects us from that which it used to be. That's science fiction. That's pure imagination. No, the reason why. Right, go ahead. Yeah.
No, that's funny. I mean, it's where. Why did it switch sides? Like
right, it's switched sides. It's like a robber breaks into my house, and then I wake up in the middle of the night. Hey, what's going on, buddy? And he ends up liking me. He's like, I think I'm going to move in. And then he ends up, working in my house to fight off, future robbers that try and break in. I mean, it's really just kind of bizarre. And I've been debating this.
Now,
go ahead.
I'm sorry. We must have a delay because I think I'm really interrupting you a lot. Sorry.
No, you're doing great.
I just wanted to know what in that article that you read about the title that you read about switching sides.
Yeah.
Do they talk about how it could have happened, or do they have? I mean, they may have made up a story, but I mean, did they give any scientific evidence for the switching of sides?
Very good question. You're asking great questions. And I do admittedly talk kind of fast. So I don't think there's a delay. Anytime you got to sneak a question in, just do it. I am completely fine with that. So the first thing I want to say to answer that really good question, Rebekah, is a lot of the critics, they'll say, well, these functions, because remember, they're just so biased. They so badly want to believe that banana plants and whales are related. So, they can't give any credit to the creationists. So they'll say, well, These were expected. These functional roles that are essential, as in we wouldn't exist without them, these were expected, they'll say, right? Which is just pseudoscientific and ignorant. Okay, I've got papers here, for example, unexpected, not expected, right here. Here's right from the scientific literature. I've got this underlined. Recent studies also point towards an until now, what does it say? Expected?
Unexpected.
No, unexpected role of the immune system for the control, for the immune system. They function in our innate immune systems. Without these ERV sequences, we wouldn't survive. We wouldn't even last 2 or 3 generations, since we're always bombarded with pathogens, bacteria, viruses. For the control of these elements, even those that do not contain fully infectious copies. I've done about 100 formal debates, Rebekah, including debates with PhD scientists, so on. A popular YouTuber named Conspiracy Cats. I debated him twice. And, one of his specialties is endogenous retroviruses. And, their responses to my basic question, Rebekah. So I ask this question to any evolutionist. Say, show me one single technical paper that demonstrates a non-functional endogenous retroviral-like element or property going from non-functional to something critically functional in the genome, such as embryological development, functioning in the immune system, determining cell types. They can't give you that evidence. It's never been observed. They have to appeal to the distant past. For example, Speed of Sound cracks me up. I love the guy, but he makes a great comedian. He always says the same thing over the last several years. Well, if we could see that, then sure, I'd present it to you. But, these functions were co-opted or adopted millions of years ago into the past. Okay, fine. Believe it then, evolutionists. It's not science. Okay, go start your own private schools, and you can teach students who are willing to learn it that non-functional retroviral-like sequences somehow became functional to the point where without them we couldn't exist. No, they don't have the evidence. It's pure imagination. And when they say it's expected, here's another one. It's all throughout the literature. This is 2022. Rebekah, I'll read this one and then I'll let you ask some more questions. 2022. Emerging roles for endogenous retroviruses in immune, notice this, epigenetic regulation. So the epigenome is this extra, I've heard you talk about the epigenome. It's this extra layer of incredibly complicated genetic information. Okay, it's above and beyond the genetic code, essentially. And we have millions of genetic switches that can be turned on or off via environment. And the epigenome is like this massive control machine that can determine, the turning on and turning off of various genes. Because if every single gene was turned on at the same time, that'd be bad, immediate death. No, there has to be some kind of control. This gene turned off, this gene turned on, especially in the developmental stages. I mean, there's certain genes that are on, they're functioning, and then once they do their job, they're turned off. If they're not turned off, then that would be bad regulation. That could lead to disease and harm. And it turns out that ERV sequences even function in epigenetic regulation. I mean, is this not amazing? It's science fiction. It's amazing. So what this says is unbiased analyses of these genomic maps have uncovered endogenous retroviruses as a what? Unexpected.
I can't read it.
Yeah, that's probably small. An unexpected ally. Oh, ally. Oh, that's funny. So they invaded. They got passed on vertically, and then they decided to work for us. We gave them a job. We're paying them the big bucks in the regulation of human immune systems. These evolutionists, I find, I'm sorry if I get aggressive with them, but I enjoy it. They're very unsophisticated. Why do we share these with all forms of life? You know what? I wouldn't be surprised if every single form of life had ERV sequences, because guess what? They function in the embryo. They function in the immune system. Life requires immune systems. And so if we have DNA elements that function in the immune system, then of course we're going to share them across the biological world. Does that kind of make sense?
Yeah. Now, is it that the fixed ERVs, do they all have functions, and then do the unfixed all lack function?
That's a great question. And that is, I think, where we are in the beginning stages of understanding the DNA language. Okay, so the gold standard of science is to make testable and falsifiable predictions. We make a testable prediction that only future observations can confirm to be true or false. Okay, I've heard some people, some creationists actually say, you know, a bad argument creationists use are those that say young earth creation is the gold standard of science. Nobody said that. Nobody said that. I challenge anybody to show a citation of that. No, we're saying the gold standard of science is to make testable predictions, and it is the young earth creationists that are making testable predictions. Therefore, we are doing the gold standard of science. Big difference, right? So this is what the critics do, is misrepresent. So what I can do then is make a testable prediction that we don't really have the answer to currently, Rebekah. So I would predict, and I do in the book, I would predict that the unfixed ERV sequences, tell me if this makes sense, I would predict that since they are relics, I guess you could say, of exogenous retrovirus, the retroviruses that got past our protectors, I would predict that they're probably disease causing, they're probably harmful, and definitely not functional in the sense that we require them to live. That's my prediction. Okay, remember, these papers, if you read them, they will constantly say, you know, we are just understanding how these ERV sequences function. There's so much more we have to learn. And that's true. We're in the beginning. It's like putting a kid under the hood of a car, a 5-year-old, and asking them, tell me what all this does. And that's intimidating. They're not really going to know much of what goes on under the hood of a car to control the car. And that's kind of how it is with scientists currently, is they really just understand so little of the DNA language. So yeah, that would be my prediction, that the fixed sequences are functional, essential, critical. And the unfixed sequences are probably harmful, but definitely not essential. And you know, that's a testable prediction that can be tested. Hey, maybe I'm wrong. Maybe I'm wrong, but that's science. But maybe that's right.
Well, let's find out.
Let's find out.
I guess
that's
we'll
what
find
it's about,
out.
right? Now, one thing I want to say is two things. There's like 20 things on my mind. One thing I want to say to help people understand is, you've heard me talk about this before. There's two really basic but important assumptions in both the evolutionary model and the creation model as it applies to genetic variation. The evolutionists, they assume that what? They assume that the origin for nearly all genetic variation is mutations, right? You understand this, that mutations accumulate, selection can utilize the genetic variation created through mutations that will lead to novel change. That's what the evolutionist believes, pretty much. Creationists, though, we don't believe that the vast majority of genetic variation within species, across species, is a result of deleterious mutation accumulation. That's science fiction. We believe that the majority of genetic variation was front-loaded, created. I don't know if you've heard the phrase created nuclear heterozygosity. It's a fancy way of saying design diversity, right? So Adam and Eve, the original created kinds, they would have been front-loaded with massive levels of heterozygosity, a state of DNA diversity that natural selection can now call upon and utilize, right? Or sexual recombination, where you get these pre-existing variants that, can be scrambled, leading to novel variation generation after generation. That's what we would expect. So where the evolutionist says this is a mutation, Rebekah, that's their assumption. It might not be a mutation. It may actually be designed. It may be a designed allele. Does that kind of make sense? Have you heard me talk about that before?
Yep.
Okay. And that also applies to the ERVs. They assume every single ERV sequence, including the fixed ones, fixed and unfixed, are the ancient remnants of past viral infections. It's a false assumption. Now, this is a pretty obvious prediction that we can make, right? If what I'm saying is true, that the vast majority of DNA variation is created, what would we predict? Well, we would predict that the vast majority of the genome would be functional. And if you notice this, the entire junk DNA paradigm overturned. We now know that, what was once believed to be junk DNA, pseudogenes, ERV sequences, ELU sequences, introns, so on and so forth, they all function in many important ways. ERVs, antiviral function, tumor suppression, gene regulation, intron cell stress responses. They allow for alternative splicing. So the point I'm trying to make, it's the creationists that are making the testable predictions, and we're making them on DNA function, but it's the evolutionists that believe that the majority of the genome is junk. And so they don't actually expect function. Picture this. I'd be curious as to your thoughts on this, Rebekah. For the most part, we're actually accidentally discovering these functional roles for ERVs. Accidentally. Could you imagine if these scientists actually had the design mindset they believed in a designer, that what we would expect is treasure versus junk? Don't you think we'd then find even more? If we're just accidentally finding these functions, if we're actually intentionally looking for them, don't you think we'd find more?
Yeah.
Okay, here's the, unless you have another question, there's right here. Okay. So I want to read this excerpt. I think this is important. Why would a creator produce functional genomic features that so closely resemble an endogenized retrovirus, right? Good question. Again, insight from the work on the KORV addresses this question. That's the koala bear species. They have what appears to be endogenous retroviruses that are really, really harming them. The structural and functional features of the pre-existing, so notice this, the pre-existing ERVs, that would be what I'm saying is the fixed ones. The pre-existing ones, the fixed ones, the ones that even the evolutionists would admit we've never observed to be inserted. It's assumed. Their capacity to copy themselves and move through the genomes. Okay, that's another important part. I'm not sure if you know that a lot of these retrotransposons, they actually have a gene promoter in them, and they actually are able to, through what's called cut and paste or copy and paste, they're actually able to move around the genome, turning genes on and off, invoking function, generating variation. That's pretty cool. Are precisely what these ERV sequences are so useful. Their capacity for retrotranspositioning, that's a fancy way of saying what I just said. They can move around the genome. Affords these sequences the means to what? Notice this. Disrupt the endogenization process of invading retroviruses. Okay, I got to break this down. This is amazing. So if we do get a retrovirus that invades and actually is passed on vertically to become an unfixed endogenous retrovirus, the pre-existing fixed ERVs, Rebekah, they can actually function by disrupting the endogenization process of these invading retroviruses. They can actually put a halt to the endogenization process, which could lead to harm. Again, it's just function after function, and many of these functions are predicated, again, I can't see the chat, but I'm guessing most of the arguments that I've seen, Rebekah, in response to the book is, well, why do they look like retroviruses? Why do they have these similarities to retroviruses? Why do they have the enzymes, integrase and reverse transcriptase? Well, that's because the way that they look, it's necessary. It's required for their functions. Does that kind of make sense? And if there's any other questions, go ahead. This is heavy. I understand. Yeah. This is heavy. Well, but remember, they believe this is their single best evidence.
Well, speaking of responses to your book, I watched Dr. Dan's response to your book. And so I wrote down a couple of things. I feel like you've answered some of those. Right. But he said that ERVs are like a giant billboard screaming common descent. And he said what you need to answer is why across lineages are you able to recover nested hierarchies based on the sequences themselves? So Do you want to respond to that, Donny?
Sure. Yeah. Yeah. So, Dr. Dan Stern Cardinal, he's responded, and he's done a lot of dodging. I've put out a Patreon article responding to some of his arguments, and a lot of the debates I've done since the release of my book, my interlocutors have utilized his arguments. So I'm more than familiar with them, and they're unimpressive to say the least. So firstly, he's assuming, again, that these are the ancient remnants of past viral infections. Never once, and you watched the review, so correct me if I'm wrong, never once did he actually show a single technical paper where retroviral-like sequences went from non-functional or even altered their pre-existing functions to become functional in the number of things we've been talking about. Never once did he answer that question. He actually hand-waved away the arguments from function, because here's the thing, the evolutionists, even the PhDs like Dan here, they don't have an answer. There is no answer for how these retroviral-like sequences of DNA can be functional to the point where if we don't have them, we wouldn't exist. Okay, that's amazing. That's amazing. And these responses from people like Dan, they are what I call a gift to creation science. It actually shows and demonstrates that in 2022, it is an amazing time to be a young earth creationist, and it's a very bad time to be an evolutionist. So what he's talking about in terms of nested hierarchies, okay? We talked about this at the beginning, right? These groups within groups patterns. We share more. So notice this on screen. I got a visual here.
Right.
So humans, chimpanzees, mouse, okay? These three, all mammals, right? Lizards, salamanders, fish. You know, you can think of the acronyms. Everybody knows, have probably heard basic taxonomy in terms of kingdom, phylum, class, order, family, genus, species, right? And the way that evolutionists believe that evolutionary history has occurred, in its most basic form, you can remember the acronym FARM. This is how I've always remembered it. Fish, amphibian, reptile, mammal. We're at the top, right? Mammals can be grouped together, right? Humans and chimpanzees share more in terms of anatomy, morphology, physiology, and genetics than a human and a mouse, or a human and a lizard, right? Just standing back, you could probably expect that, right? Obviously, we're going to share more with a chimpanzee than we do with a fish. I mean, it's really not that complicated. But here's the thing. Humans and chimpanzees can now be grouped into, based on similarities, grouped into the category primates or great apes. We can all be grouped together, even with the mouse and the lizard as vertebrates, and so on. It's just these groups within groups patterns. I mean, you know what? I don't think the chimpanzee really cares where we classify it. Okay. There's a lot of people out there that have a lot of time on their hands. They like to classify. We classify things all the time. Go to a car lot where you got 500 different cars. You have the SUVs in one section, the vans in one section. You got the sedans, the hatchbacks. We always group things. Your work shed, you group your tools, your kitchen drawer, your forks, knives, spoons, whatever. It's just classification systematic. It's not proving anything.
Right.
So here's the thing, Rebekah, based on everything we've been talking about, okay, watch how easy this answer is going to be for you. So if the majority of endogenous retrovirus-like sequences are functional and therefore created units of DNA function, do you think that we would share more of these with the chimpanzee than we do with the lizard?
Absolutely. Yeah.
Evolution.
And that's why when I saw this question, even, it seems to assume that we are recovering lineages. So it says, why across lineages are you able to recover nested hierarchies? But that assumes that there was a common ancestor, right? But we're not starting with that assumption. We're starting with the assumption that we have similarities with certain animals. That doesn't mean common descent. It just means that God created some creatures to be more like us. So yeah,
that is such a great point you make. They admit, for sake of time, I won't do it, but I got a whole, I got 1,000 slideshows, and anybody who ever wants like my slides, let me know. I'll send it to you. I've put a lot of work into these. Here's the thing. The technical papers from the secular literature admit that they assume a tree. When they go into building their phylogenetic trees, their family trees, they assume there's a tree in the first place. They assume whatever organisms they are, putting, if they take a genetic sequence, put it into the computer, and it shoots out, a manifestation in the form of a tree. Okay. Let's say they take the DNA sequence from humans, chimpanzees, orangutans, gorillas, bonobos. Yeah, the computer will spit out what looks to be like a relationship, right? Some hierarchy within the great ape family. They're assuming that exists in the first place. They're assuming that these creatures are related in the first place. Well, here's the thing. Yeah, we all have DNA, RNA, proteins, right? We, especially the great apes, we have binocular vision. We've got, 2 hands. We need to walk around. We have 2 legs, 2 feet. We have hair. Of course, there's going to be similarities. So notice this. It just so turns out, Rebekah, and here's why I want to lay down some context in its most basic form, okay? Here's the model that I present. What does the Bible say in Genesis? The Bible says that we, as humans, are created in the image of God, correct? Yes. So therefore, I would predict that there must be something about us that reflects the divine. So when it comes to the question of ancestry and the creation versus evolution debate, maybe, just maybe, we could be able to get a sense for how God designed the biological world and the patterns that he used based on the way we design, right? Does that make sense? Well, it just so turns out that we as human engineers design in what? Nested hierarchical patterns. We design in what? Homologous patterns. The exact same patterns we see in the biological world. Isn't that amazing? Notice this. Here's a hierarchy. A sedan can be grouped in with vehicles with 4 wheels. Which vehicles do you know have 4 wheels? SUVs, crossover vehicles, sedans, hatchbacks, vans. You can group them together with 4 wheels. Land-based vehicles. Oh, wow. I mean, you're going to be bringing in a ton, right? Engine-powered vehicles. Well, now you're bringing in the airplane, the boats. And it just so turns out the sedan, I'll ask you, it's more rhetorical, but Rebekah, does a sedan share more with an SUV than it does with a bike?
Yes.
Yeah, that's a hierarchy. We share more with the chimpanzee than we do with a fish. Ooh, we share more with the chimpanzee than we do with the snail. Does that mean humans and snails are related through common ancestry? That's the evolution debate challenge that we've been doing in 2022. And the best they got is ERV sequences. And ERV sequences fail. Okay, this is a hierarchy. It's not evidence for evolution. And I think you know exactly what I'm saying here. Any points you wanted to add to this one?
Well, no. I was just going to go on to talking about another thing, which is hotspots, because this is something Dr. Dan mentioned. He was talking about hotspots and how, like, you presented that, hey, we see mutations in these hotspots. So it's like, I don't want to misrepresent you because I'm just kind of paraphrasing or, taking an idea. But so, like, can you explain what Dr. Dan's problem with your discussion of hotspots is that it has to do with the sequencing. Like, it wouldn't, like, yes, we can kind of predict where hotspots are, but that doesn't tell us, that doesn't explain why the sequences are exactly the same. Can you talk about on that topic?
Absolutely. And I just pulled up from my phone the chat just to see the triggered atheist. Creation Myth says, I don't think SFT understand the question. No, I don't think Creation Myth, aka Dodgeball Dan, understands just how badly his ERV argument has been demolished. Okay. But, I don't put too much confidence in those that believe there are 25 genders. But anyways, that's besides the point. Okay. So when it comes to the mutational hotspots argument, right? Firstly, Rebekah, if we go back a little bit, to when we started this conversation, right? What was one of the major differing assumptions between the creation model and the evolution model? Well, we would explain the vast majority of genetic variation as being the result of what? Created nuclear heterozygosity, right? Makes sense. The evolutionists would explain the vast majority of genetic variation as being the result of mutations. So in the book, I have an entire section that deals with this question. What they assume is a shared mutation, okay, may not actually be a mutation. It may just reflect the functional requirements between the respective organisms. For example, a chimpanzee and a human are going to utilize their ERV sequences slightly different, right? Information flow between a human and a chimpanzee are going to be different. So therefore, DNA differences that are assumed to be mutations may actually just reflect functional differences. And that goes for the mouse, right? An ERV sequence in a mouse that functions in embryological development may actually exist for embryological development, but it may be utilized different in the mouse because there are differences in the mouse between the human in terms of morphology, anatomy, physiology, and genetics. Okay. So firstly, does that kind of make sense?
Yep.
Okay.
Absolutely.
Okay. So, and that's what I would predict is the more sequencing we do in terms of genomic functionality, right? We can do mutagenesis. We can knock out snips of DNA. We can see if there's an issue, if there's a disease. There's many different ways we can determine what the function is or if it is functional, right? These are tests that have not yet been performed, especially on humans. It's premature. Genetic knockout test, that's the gold standard of genetic function tests. So the point I wanted to make there is there was no engagement from Dan on that. Here's an analogy I want to use, and then I'm going to go to the mutational hotspots. And the reason why I'm talking about this is because my main argument anyways for shared mutations in the ERV sequences is the fact that what they say is a mutation, there's a strong chance isn't even a mutation. Remember, they assume that all of these SNPs, single nucleotide polymorphisms, you'll see Dan in the chat say SNPs, right? SNPs. They'll assume those are the result of mutations because that's their evolutionary bias. That's what they believe. That's their starting point. Where we would say, no, the difference that they say is a mutation might actually be what's called a created difference. And that's one of my main arguments on the shared mutational argument that form hierarchies. But another thing that we know is that a lot of genetic sequences and DNA elements, they actually have multiple functions. And this is a really fascinating point that I do want to at least show you before I move on. Protein moonlighting, for example. Notice this. A single protein with multiple functions might seem surprising. Again, it's always a surprise to the evolutionists, right? Because they expect junk, not treasure. We expect treasure, not junk. So it's not surprising to us. But there are actually many cases of proteins that moonlight or have more than one role in an organism. What that means is you have one protein, Rebekah, but it functions in many different ways. So you might have a protein, a nuclear DNA protein in a chimpanzee and a human. Okay. You can even say that it's shared, but it actually has multiple functions. So its primary function might be the same in a human and a chimpanzee, but its secondary function could be different because the organism is different. And that could be the same thing with the ERV sequences. Okay, maybe they look like they have shared mutations. Maybe they're in the same spot, but their primary function and secondary function, primary function might be the same, functions in determining cell types, but its secondary function could be different. And again, this is a future testable prediction that I didn't see Dan really engage. Okay,
now
go ahead.
Are you saying
I'll get to the hotspots one soon, but go ahead.
That's okay. That's what I was going to say. I'm just let me ask this question before you move on. Are you saying that some of these things that maybe we're considering hotspots are actually not even mutations? Like they're not actually hotspots?
Great question. Great question. So it's not that the hotspot, so the mutation is different than the hotspot, and I'll explain. What I'm saying is the mutation itself might not be a mutation. Okay. Let's say they hired me Let's say they hired me to, well, let's say do you speak any Russian? Guessing not.
No.
So let's say they hired you to go to Russia, move to Russia to become a proofreader. So they give you a book, it's 500 pages, and they want you to read through it. It's written in Russian. They want you to read through it and pick out mistakes, typos. You're a proofreader, you're editing it. How good of a job do you think you're going to do? Probably not a very good job, right? Terrible. You're not going to know what is a mistake and what's not. A sentence, since you don't really understand or read Russian, might look normal, even though there's a few mistakes in there. That's how it is right now with the DNA language. Scientists assume everything's a mistake, when in fact, it may not be a mistake. We see that. A prime example is in pseudogenes. For years and years, their main argument was, well, we have these shared pseudogenes, these shared genetic mistakes between humans and chimpanzees. They were inherited from past common ancestors. Okay, it's junk, evolutionary leftovers. It's a mistake. Now we know, and I could show you, 20 papers at least, where pseudogenes are actually necessary to sustain healthy life processes in the cell. Evolutionists assume they're genetic mistakes. No, they're genetic, they're treasure in the genome. They're functional. They're not a mistake. That's how it is in the DNA language, where they say, oh, here's a mutation in the ERV sequence. It's shared between humans and chimpanzees. Oh, they must have inherited that. No, they don't know that. They're so ignorant of the genome right now. Okay, we're understanding that the genome is 4-dimensional. It's nested. It's filled with multiple overlapping codes. You got a gene over here on this sequence, a gene over here. They're interacting with each other. They're working together to control regulation and gene expression. I mean, it really is science fiction on the molecular level. So that's what I'm saying, Rebekah. But when it comes to the mutational hotspot argument, I'm saying that some could very well be mutations. But we know, and here's a few papers right here, if my screen's still shared, 1, 2, 3 at least, and I've got a bunch in the book, that talk about how the evolutionary community used to assume, for the most part, that mutations are random. They just occur anywhere, and they kind of need this, right? To take your single-celled like ancestor into a whale over billions of years requires almost limitless changes, right? So to them, the mutations are random, they hit wherever, and then selection can act upon them. Well, there's a ton of papers right now in the literature that are showing, and I'm going to show some papers on it here, that demonstrate that there's spots in the genome that are what's called hot spots. They're more likely, does this make sense? They're more likely to be hit by mutation. So the mutations are not necessarily hitting in just random spots. And there's one unexpected, okay, right here. So notice this. Scientists are admitting more and more that mutations are not entirely random. They acknowledge these findings. Here's an excerpt. Mutations occur when DNA is damaged and left unrepaired, creating a new variation. The scientists wanted to know if mutation was purely random or something deeper. What they found was unexpected. We always thought, notice this, we always thought of mutation as basically random across the genome, says Gray Monroe. I believe he's the lead author of this paper, not a Young Earth Creationist paper. He says, it turns out that mutation is very non-random, and it's non-random in a way that benefits the plant. It's a totally new way of thinking about mutation. Now, I'm going to have to spend a couple of minutes here because I've watched Dan's review and I know what his response is, okay? So firstly, Dan's going to say that if these ERV sequences are functional, or as functional as I say, let's say in the embryological development, if a mutation, let's say it's a hot spot for mutations, a mutation hits it. Well, Dan would say, and on the surface level, it seems like it would make sense, it would kill the organism. Because if the mutation hits an essential, a critical functional gene or DNA element, then the organism will die. It won't be able to pass on its genes. No kids, right? But here's the thing. Most mutations are actually what's called effectively neutral, nearly neutral. That means ERV sequences can be hit by mutations, but those mutations are not necessarily going to kill the organism because they might only be slightly damaging. And so, yes, they can build up. Let's say hypothetically you have, the chimpanzee and the human. They both already look very similar in terms of anatomy, morphology, physiology, genetics. And then they have ERV sequences that function in the embryo. Well, guess what? We live in the same world. We live in a lot of the same environments. We consume. So there's a lot of similarities. Therefore, we may be subject to the similar mutations. Okay. And again, this goes back to our design hypothesis when it comes to designed means like modes of transportation. You can just research what areas of your automobile are most affected by corrosion and rust. All automobiles, for the most part, there's actually a few different parts of the car that are more so affected. There's hot spots, even in terms of rust. Like your engine is subject to corrosion, to damage. But guess what? The first rust molecule that builds on the car, on the engine, does that kill the car? Does the car stop working? No. It takes a while. They build up, and then eventually, the engine is trash pretty much, and the car is useless. Okay. So no, in the same way that we have hot spots in various modes of transportation, we also have hot spots within the genomes of living organisms. Here's the last thing I'll say as a rebuttal to what Dan would say. Dan would say there's not as many hot spots that a creationist would need. But again, this is assuming that the vast majority of the genome is non-functional evolutionary leftovers. If the majority of the genome, as the evidence suggests, is active and therefore probably functional, including these DNA elements like ERV sequences, then guess what? There are going to be many more hotspots than the evolutionists assume. Over and over again, what do they say? This is surprising. This is unexpected. The evolutionists, like Dan, they're premature with their arguments. Give it another 20 years. Their arguments they're using now, they're going to be completely overturned. Ask them a question. Have you ever predicted, before we found the functions for ERV sequences, did you predict these functional roles? And the answer is no, they didn't. What they do is they post hoc, ad hoc rationalize away the evidence. They force fit it in rather than admit defeat. I understand that was very long-winded, Rebekah, but if you want, go ahead, play devil's advocate. Any clarifying questions? That's what I'm here for.
Well, I mean, Dr. Dan is in the chat and he's saying it's not his argument. But I kind of feel like Dr. Dan, I think I sent you an email last night. Maybe I didn't finish it, but I would love to have you on and you can explain your argument more clearly, and we can respond to that. So
here's one thing I didn't mention. He would say that it's these hot spots are more probabilistic rather than deterministic. But again, okay, he's partly right. But if the majority of the genome is functional, in the same way you have a car, where all the parts for the most part are there for a reason, they're there for a purpose. That means you can have totally unrelated cars from totally different manufacturing companies, okay, that can be hit with the same problems. Take your car to a mechanic. They'll say, what's going on, with your car? What's the issue? Well, it's making this funny noise and, there's something going on over here. Right away it clicks. Oh, that's what's going on. Because all modes of transportation have similar problems. Doesn't mean they all evolved from a skateboard. Okay. If the vast majority of the genome is functional, right? And you have phenotype and you have the genotype and whatever, a lot of the physical features are being coded by the underlying genetics. Then that means various unrelated creatures are going to be hit with the same problems. They're going to be hit with the same types of mutations. I mean, to me, it's pretty basic, but here's the thing. The Bible says that there's scoffers who are willingly ignorant. I'm not really trying to, it'd be nice that a lot of the more militant evolutionists would come to repentance. But at the end of the day, yeah, we understand there are those that are willingly ignorant. This is a perfectly reasonable answer. It doesn't mean that they're going to tap out. Okay, so go ahead, Rebekah.
Well, and how do these hotspots relate back to endogenous retroviruses? Are you, because I is it also equally likely for endogenous or exogenous retroviruses to become endogenous retroviruses at the same places?
That's a good question.
Does the hotspot apply to that?
So the reason why the hotspots are important is because we have the ERV sequences, right? They are shared between humans, chimpanzees, orangutans, gorillas, so on and so forth, all throughout the primates and the vertebrates. They form hierarchies, but this is a multi-layered argument from the evolutionists. We also have what looks like, and I think you know why I say looks like, because they assume it's a mutation, where I'm saying it might not be. They see what looks like to be shared mutations. Okay. So tell me if this makes sense. You have an ERV sequence that is shared and forms a hierarchy, but we also have within the ERV sequence mutations that are shared. So we have this.
Now
right?
I'm making the connection.
Yeah.
Okay.
So it's like a double argument. But most evolutionists won't even bring up the second part to the argument. Only the informed ones like Dan, and credit where credit is due. Dan's probably the best of the best, you know, and him and I do get passionate, but you know, I do respect his knowledge for sure. And I've learned a lot from the critics, engaging them. Okay. I just don't know if Dan is entirely even open or objective on this. Okay. Which is fine. So they'll take that second argument. It's like their argument is like a hierarchy too. Arguments within arguments.
Okay.
So the ERV is shared, but then it looks like we have mutations within the ERV that's also shared. So what that means to the evolutionists is, okay, we were hit with an exogenous retrovirus. It was passed on. It became endogenous, and it got hit with mutations. Okay. And
now,
then
how do we know those are mutations?
Exactly. It's literally what they say is a mutation is my analogy earlier, Rebekah. If you were a proofreader
about
in
how
Russia,
different expressions. Yeah.
Well, no, if you look at like a single nucleotide polymer, let's just look at like a base substitution. You swap one nucleotide for another, a DNA letter. Okay, so you're comparing genomes, and they're assuming evolution, common ancestry. So they say, well, you know, this DNA difference here between the humans and chimps, since humans and chimps shared a common ancestor, must be due to a mutation. That's circular. They're assuming evolution to say this is a mutation. They don't know that. If we have, here's one thing I'll say. If I'll use the Y chromosome as an example. The Y chromosome is inherited from our fathers, right? It's passed down uniparentally. And every single human male Y chromosome is nearly identical, 99.99% the same. But there are what's called geographically specific Y chromosomes. So you have, you know, Asian Y chromosomes, African Y chromosomes, European Y chromosomes. You want to know why? Because as people groups spread out, they become isolated. There's no reproduction, you know, between some people in Africa and some people over in, I don't know, Canada. So each group accumulates their own mutations, right? That is pretty easy to determine. Okay, that's a mutation because it's just exists in a small population. And a lot of the times these mutations are disease causing. So it's like, okay, that's probably a mutation. But if you're looking at a DNA difference that for the most part, every single human being has, and maybe it's functional and not disease causing, then we as a creationist, we could say, no, that's probably created. The evolutionist is still going to say it's a mutation. That's their basic assumption, right? So it's a battle of worldviews. And Dan's arguments, unfortunately, and I wish he would take a different approach. This is why, to me, it's unsatisfied. He's assuming evolution and common ancestry to argue against my arguments. He's trying to say, oh, you didn't explain the shared mutation argument. Firstly, we have to determine if it really is a mutation that we're looking at. He doesn't know. This so-called mutation occurred millions of years ago in a hypothetical common ancestor that was then inherited. No, this so-called shared mutation may actually be a design difference that specifies various functional roles in the creatures, right? Like a truck, an SUV, and a sedan.
Yeah.
They could have a feature that's exactly the same, but it could function slightly different depending on what the vehicle is. Like a truck, you know, it could be built with whatever certain ingredients, you could say, but still function totally differently. So, there's a lot. I want to go over this slide because I believe this is Dr. Kevin Anderson. I think he breaks it down nicely. And this is a really technical topic. So I do want to go over this and see if it kind of enlightens some people that are kind of like, what are they even talking about? So it says also, since the DNA location of many mutations is not random, right? This challenges an assumption frequently made by evolutionary biologists. When two organisms share nearly identical mutations, this is what Dan is saying. Oh, the chimpanzee and the human, they share identical mutations. So they must have inherited it from a common ancestor. Biologists often assume this is evidence of shared evolutionary history, right? The identical mutations must have been inherited from a common ancestor, just what I said. The shared mutational pattern presumably shows a Darwinian lineage. Notice this part. However, this reasoning assumes mutations are randomly scattered throughout the DNA. Thus, the only way two organisms can share identical mutations is through shared ancestry. Yet, because many mutations are not randomly located, shared mutations could result from shared mutational hotspots rather than shared evolutionary history. So I'm going to break it down like this, Rebekah. If we have an ERV sequence in a human and a chimpanzee and it functions in embryological development, well, then guess what? That ERV sequence is probably also going to be subject to the same mutations, the same kind of damage. And if the majority of the genome's functional, that means there's even more hotspots than somebody like Dan would want to say. Dan would say, yes, hotspots exist, but not nearly as many as, Standing for Truth would need. Or he thinks that it's more so probabilistic rather than deterministic. But no, if design diversity is true and God front-loaded living organisms with various classes of functional DNA elements, then no. In a way, a lot of the mutagenic discrepancies could be determined because God determined this sequence functions this way. And so a problem that would arise could, in a way, be determined, right? This is a heavy topic, I will say, but I hope some of that made sense, Rebekah.
Yeah, this has been incredibly helpful, and I know we have to wrap it up. So do you have any, are we just getting started?
I've been screen shared the whole time, actually. How much time are we on?
Ooh, well, I think we said So do you have final thoughts, or are we missing a big chunk of what you wanted to share? Or?
Let me look through the chat, see if there's any arguments that, so again, just looking at the comments, Dan, it's all about the mutational hotspots. But again, my argument is not only mutational. Yes, mutational hotspots exist. Okay. For example, we have what's called, I don't know if you've ever heard the Gulo pseudogene. Gutsy Gibbon loves this one. Okay. We have a so-called broken gene in humans and chimpanzees. We can no longer synthesize our own vitamin C. And it looks like
yes. I wanted to ask you about this. So good. I'm glad you're talking about this.
Well, it kind of applies. And you know what? Okay. I just finally gave you the chance to not screen share, but I'll have to screen share again because I do have slides on this. So appreciate your patience.
Yeah.
Okay, so let me find it. I did a whole video actually on Gulo addressing Erica's argument, and there's been no response. But so as I find it here, there's certain genes. Oh, here it is. Okay. So the Gulo and genetic degeneration. So what we actually find here is, notice this part. Thus, degradation of the Gulo gene in a wide array of mammals and birds is a relatively common occurrence. So when it comes to a gene that assists in vitamin C synthesis, for example, well, there's a lot of food, right? A lot of fruits, for example, where we can get vitamin C just by eating. Especially today, I mean, you take vitamin C supplements. So here's the thing: that gene, although functional, it's less likely to be preserved by natural selection. because at the same time it's synthesizing vitamin C, but you know what? We're getting it by other means. Okay. So notice this: loss of vitamin C pathway function in gulo gene degradation has also been detected in guinea pigs, great apes, and humans. In addition, naturally occurring scurvy and osteogenic disease pathologies related to gulo inactivating mutations and large-scale deletions has been documented in rats, mice, and pigs. So the point is, even what the evolutionists would say is a more distantly related ancestor, right? They would say we split from a common ancestor with chimpanzees way more recently than, let's say, with a rat or a pig. Yet we actually see that this so-called gene, it breaks down and it's hit with mutations independently of common ancestry because it's more susceptible to mutation. It's more likely to be hit by a mutation. So there's one thing that I wanted to, okay, so right here.
I have a question about that. So in the animals that we see the degradation of that gene, is it across the entire, like, is it for the entire species? So like you mentioned guinea pigs. So do all guinea pigs have that problem? And do all, these other animal species have that problem? Or is it kind of like working its way in?
Good question. No, it's widespread. It would have already happened. From my understanding, and I'm open to correction on this. This wasn't observed either. This is assumed. There's actually some creationists that would say the gulo gene, and like Dr. John Sanford and Christopher Rube, I believe, make this case in Contested Bones, that what the evolutionist is assuming is a gene deletion may not be. Again, it's their assumption of mutations. What they believe is a gene for vitamin C synthesis in humans may actually just function in a totally different way that we don't necessarily understand. From our model, it kind of makes sense. Adam and Eve exist in a garden, and we're commanded to eat what? The fruits, the vegetables, and the seeds. That's a lot of vitamin C, right? So maybe God intended our so-called gulo pseudogene, to be utilized in a different way than chimpanzees. And you know what? That's reasonable. That's a prediction. That's a testable prediction. We don't really have an answer for that yet, but that's a testable prediction in print. And I'm not going to totally disregard that. That could be the case. But even if it was a mutation, notice this. This indicates that the gene, the gulo gene, may be predisposed to the effects of genetic entropy. Because guess what? It occurs frequently in mammals and birds. Gulo gene dysfunction appears to be a widespread. So here's kind of an answer to your question. A widespread feature among not just humans, apes, and guinea pigs, but terrestrial animal life in general. Taxonomically restricted and do not. So that would mean taxonomically restricted. This gene has been broken independently in different lineages. As in, it didn't necessarily break in a distant common ancestor and then was passed on. No, it is a gene that is more susceptible. And what's interesting is that there's actually some discrepancies. So right here is kind of technical, but I think Dr. Tompkins, he's a PhD geneticist, he makes a really good point here. He says these exon deletion events are taxonomically restricted in humans and in each of the different ape species tested, and in combination with an extreme lack of sequence similarity. So what he's trying to say is the sequence within the gulo locus, or location, the genetic sequence in primates, it actually has, to a degree, a lack of sequence similarity that the evolutionists would have to, again, post hoc, ad hoc. They'll have to say, well, since the split, there's been some changes. So it's not nearly identical. There's been changes since the split. But again, that's circular. They're assuming common ancestry to prove common ancestry. That's a logical fallacy. And that's what I'm seeing with this shared mutation argument, Rebekah, is it's always, explain the shared mutations. It's like, firstly, we need to determine, is this even a mutation we're looking at? Because there's a good chance that it's not, right? So there's a lot there. Well, hey, look, we even got the gulo pseudogene one, and that's another one of their favorite arguments. But that is a clear example of a mutational hotspot. They've lost that argument. Even if it really is a similar break, right? Here's the thing. I don't know how many cars you've owned in your life, but let's say, I've owned a Cruise, a 200, and a Charger. And you know what? It's funny because in the wintertime, they all might have trouble starting. Or heck, the air conditioners could break down in the exact same way, in the exact same spot. Does that mean that they're all related to a common Arctic ancestor? No, it just means common problems due to a common environment. That's an easy answer. But the deep technical answer is it might not even be a mutation. And that's what I'm leaning more towards. I asked Dr. Nathaniel Jensen, I say these hierarchies in endogenous retroviral sequences that cross kinds, right? Those mutations, do you think that maybe they're not mutations? Right? I wanted to make sure that my prediction here made sense. And he said, anytime that a creationist sees hierarchies in genetic markers that cross kinds, we have to first ask the question, is this a created unit of DNA function? Or is it really a mutation? That's where we need to start, Rebekah.
Right. Wow. This has been so incredible, Donny. Like, you've answered so many of my questions. I have loved this so much. And I know Dr. Dan is frustrated in the chat, but Dr. Dan, I'm looking forward to hearing your response. And he's accepted my invitation to come on. So I look forward to hearing from him. And so
Rebekah, I don't want to interrupt you.
You got more.
Yeah. Well, oh yeah. I mean, I could go all night for sure. But so what he says here, that's not a hot spot. That's a region without purifying selection. Okay. So what he's going to say is a lot of these papers on mutational hotspots, the papers point out that the regions, the genetic regions that are not being hit with mutations are functional regions. Right. So in a way, selection's preserving those regions, and the regions being hit or subject to mutations are non-functional regions that maybe reflect evolutionary leftovers. I think already, Rebekah, you can see the assumptions in that. They assume that only portions of the genome are functional and necessary, and most of it is non-functional. That doesn't make any sense. We have over 60%. We have evidence that over 60% of the genome is active. Okay, it's actually closer to 80. Somebody like Dan, and I've been debating these guys for years on this. One guy that comes to mind is CRISPR. I think he's like a cancer scientist. And 3 years ago, we were having full debates on junk DNA. I've heard it all. And they try and say, well, this activity is spurious. It's low level transcription. It's not really doing anything. And it's like that's completely ridiculous. And, for sake of time, I'll just say I did a 2-part series with Dr. Fuzz Rana, PhD biochemist, titled Refuting Junk DNA. And Dodgeball Dan, he put out a video as defending junk DNA, and we actually dealt with every single one of his arguments. So no, I can confidently say the junk DNA paradigm has been completely overturned. So if more of the genome is functional, then what does that mean? More similar spots to be hit with mutations. Just like the engines of various modes of transportation. The engine is probably the most important part of a car, right? Without the engine, you're not going anywhere. And yet the engines are hit with similar rust events, right? Corrosion acts upon similar spots in various engines. Well, what does that mean? It's a part of a car that's functional, and it's also hit with a lot of the same problems. Well, you know what? Chimpanzees and humans have a lot of functional DNA elements that serve a common purpose and also are subject to common problems. Pretty reasonable, right? Maybe not to some of the more militant evolutionists, but what are your thoughts on that?
Yeah, I mean, I've been actually wanting to ask about the vitamin C thing, so I'm glad you explained that, and I think that's a great explanation that it as like similar creatures, we have areas of that would be more likely to get changed, mutated, but also, like you said, we're not even sure that is a mutation. That could be just a difference in organisms.
That's
Right,
exactly
like-
right.
Yeah.
A simple answer, and I point this out in the book, is mutational hotspots, especially if the genome is mostly functional, which the evolutionists would reject. Then there's going to be more hotspots in the genome, which actually make mutations more deterministic rather than probabilistic, right? Because if the majority of the genome is junk, this is what I believe Dan means by probabilistic, in a way, at least a part of it. It would be like if you were parachuting off of a plane, and your goal was to parachute into North America. You know, that's the hot spot. So no analogy is perfect. So then you parachute into North America out of all the places on the earth, right? But it doesn't mean that you've parachuted into an exact location at an exact house in the address you were looking for. No, I mean, you could parachute into North America, but completely away from, you know, a determined house. Okay, that's if the majority of the genome is junk, though. The majority of the genome reflects functionality, then no, it is a lot more deterministic than it is probabilistic. So that, you know, that for the most part will be his response, is that you know, it's more probabilistic versus deterministic. I'm trying to find one more slide to really leave no stone unturned for you. So maybe as I find it, if you, yeah, let me just, I have too many slides here. So I'll just point this out in a nutshell. Okay.
Yeah.
This idea, Rebekah, that the majority of the activity in the genome is spurious or just noise and not really doing anything. It's really, really ridiculous, because we understand that, and Dr. Fuzrana pointed this out with me, to transcribe a portion of the genome, it's very, very energy intensive. Okay. It's an energy intensive process, right? Protein synthesis, DNA to RNA, right? Transcription to protein, translation. This is all a very energy intensive process. And every time you add a nucleotide to a growing RNA chain, a massive amount of energy is being consumed to do this. That's a lot of energy. That's a lot of energy being released as heat for no reason at all, to apparently create, you know, DNA elements that do nothing. No, if all this activity really was noise, all right, don't you think, Rebekah, that over time natural selection would remove this junk? Isn't that a waste? I mean, if your drawer, if your dresser in your room is just being overloaded with junk, with clothes that you no longer wear, eventually you're going to be like, listen, you know, this is a state I'm currently in. Eventually you're going to get garbage bags and put the clothes in there that you don't wear and get rid of it. It's junk. It's wasteful of energy and resources, right? You're making room for the new stuff. But yet these evolutionists are trying to say that over millions of years, selection kept around all this junk just to transcribe for no reason. I mean, does that make sense? What a waste of energy and resources, right? If it's not doing anything, get rid of it. If you don't use it, lose it. That makes sense?
Yeah, absolutely. Yeah.
You're probably, but
No,
I want to,
well,
okay, go ahead.
no, go ahead.
Well, think about this. Okay, because the evolutionists are going to say, no, it's just better for the, like, this is what Dan will say. It's just better that we transcribe the noise anyway. It's actually less work. Okay, it's post hoc. So, Rebekah, the evolutionists believe billions of years ago, from some warm pond, right, non-living chemicals somehow organized themselves into the first self-replicating molecule, the first single cell. From single cell to multi-cell to what? Farm, right? To fish, amphibian, reptile, mammal. That is massive amounts of increases in phenotypic complexity, novel body plans, novel and meaningful information, right? You're going from a single cell like ancestor to a whale through natural selection, mutations, environment, so on and so forth. So from a single cell like ancestor to a whale, think about that. But yet you're telling me the natural selection could not remove the junk. It, with mutations, took a single cell like ancestor to a whale, but just forgot how to remove junk? Give me a break. This is science fiction. This is imagination. Dan can believe in it all he wants, and he's invested. He's invested a lot of time, energy, money, teaches it. Of Of course, he's, for the most part, not going to be objective enough to agree with me. Okay. And notice this, they can't explain these functional roles, Rebekah. They say, well, it occurred in the distant past. And well, HIV is kind of selfish. HIV, this is what Dan said in his video. You probably remember if you watched it recently. HIV makes its way into a cell. And when there's enough HIV in the cell, it blocks it off. Kind of like intruders get into like a castle. And then once they've infiltrated, they take the drawbridge up. Nobody else can get in, right? So HIV does that. So he's saying, well, maybe just imagine SpongeBob style, HIV can do that, which guess what? HIV kills a person. Nobody wants HIV. Maybe because HIV could do that, the exogenous retrovirus will hit in the right spot in the same way. It'll be passed on vertically, and then it'll become a benefit to the host and act as antiviral protectors. Sure, just imagine. It's not science. Dan, go start a private school, teach that to anybody who's willing to pay. Okay. And sure, it's all good, but that's not science. It's science fiction. And that's the best that they got, Rebekah. HIV kills, but these ERV sequences actually prevent us from dying due to the intrusion of exogenous retroviruses. That's a lot different than what HIV is doing. Okay. HIV is being selfish, but it still leads to death. All right. I actually think I just dealt with all of Dan's arguments, actually. So I'm actually going to be releasing a bit of a handbook responding to his arguments, and very disappointing. So, okay, go ahead. This has been fun. I hope it hasn't been too much, though.
No, it's been great. Thank you so much. And if anybody wants to find Donny's book, the Endogenous Retrovirus Handbook, the link is in the description. And I don't think I need to promote your channel because everybody already knows the channel, but the link is in the description if this is your first time seeing Standing for Truth. So go to his channel. You'll get a lot of great content and a lot of good debates. So thank you so much for being on, Donny, and I appreciate your work. And thanks everyone who's been here.
Appreciate it, Rebekah. This has been fun. God bless.
God bless you too.